And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
You probably got about 8 seconds to say something offensive.
It's all yours, SFT.
Awesome, guys. We're here this afternoon with Adam Heap, Raw Matt, and myself, of course. We are continuing the discussion with Adam Heap. So I want to thank him for joining us this evening. If you haven't seen our first talk, more of a formal debate on modern day debate on generally the same topic of ancestry, universal common ancestry, or I guess you could call it biblical limited ancestry. I definitely recommend checking that out. This is going to be more of an informal dialogue, touching on maybe some of the loose ends or some of the topics from the first dialogue that we'd want to elaborate on more. Adam always makes for a good discussion. He's, from my understanding,
That's a lie, by the way.
Hey, you paid me to say that. Now
you can't prove that. Prove me the scientific study that says that I have a good discussion. I can't find any.
All right, I'll accept correction on that one. From my understanding, Adam's got a background in zoology. I think he's also has a background in herpetology. Actually, did you want to clarify that, Adam? Or
I have a master's degree in zoology and herpetology. Herpetology being primarily the study of reptiles and amphibians, but I amphibians are bleh. I'm more into the reptile side, me.
The cool side.
Yeah, the cool side, yeah.
Well, that being said, we always enjoy talking with Adam because he's certainly qualified and well educated, and he's looked over a lot of the material that Raw Matt and myself have put out. So he brings forth good arguments and good objections, which gets us all to think. And like I said before, it leads to a good, memorable discussion. So once again, thanks guys for joining us. I'll pass it off to Adam Heap and Raw Matt as well, if you guys want to say a couple short introductions, and then we'll jump right into a couple related topics to ancestry.
I'm happy to have my introduction as a simple yet tangential question, and that is, why is so much as community Canadian?
Oh, are you saying because your intro is going to lead in with the question? Do you want Matt?
No, that's just it. I don't have an intro, so that my intro is just a tangential question that I've always wondered. Why are there so many Canadians in this bloody community? Where do they all come from?
Where did we all come from? Canadians?
Yeah.
Canadians produce Canadians, my friend.
But at one point they weren't Canadians.
No, that's true. We could probably trace. I could probably trace my Canadianness, I guess you can call it, right back to Adam and Eve. Maybe. What do you think, Matt?
They were Canadian.
Well, you're American, Matt.
Yeah. But Canada's above us, and they're bigger, so I'll leave them alone.
Oh, I was looking on the map yesterday. I was doing some work in my brother's house. He's getting his floors done, and he's got this big picture of the map. It's not flat. Sorry, guys. But
Okay.
I was looking at the country sizes, and sometimes when you glance at it, if it's been a while, I mean, the size of Russia or the size of like Canada too, even in comparison to like the States so Matt, don't mess, brother.
That's true, but I think all those maps are fake news. So
I heard the other day that the last place on Earth to ever be discovered was actually Antarctica. Which is probably quite hard to believe, considering the tiniest little islands that you get in the Atlantic and Pacific Oceans that were found before the whole of Antarctica.
Yeah, that is interesting, actually. Well, Adam, you're always welcome to come visit in Canada. We've got really good coffee. We're pretty easygoing to talk to.
You've got to provide me a source for all this, by the way.
Okay, so I guess getting into it then, did you, just to start it off, if we're going into ancestry, and if not, not putting you on the spot, but we discussed mitochondrial Eve a great deal in the first discussion,
yep.
As well as Y-chromosome Adam. Mm-hmm. You did bring up a very interesting study done in Chinese men, I believe it was.
Yeah.
Do you want to elaborate on that, actually, just to kind of get the audience up to date?
From what I recall from that paper, it was essentially 2 Chinese men, 13 generations apart, and they measured the amount of differences in the mitochondrial DNA. So essentially, they added it all up. They saw where the common ancestor between the two was, which was about 1805, and it was a case of simple multiplication, essentially.
Was this the Y?
That's the Y chromosome. Yeah.
Right. Okay. Just clarifying.
Yeah. So that's it. From that, they got a mutation rate, which aligned with the human chint split of between 5 and 9 million years.
Right. So I think in that portion we were going over how we can look at like mitochondrial DNA, for example, and I know there's been several papers looking at that, mutation rate and coming up with a date for E, for example. We were pointing out the fact that, not only does the mitochondrial DNA go back to 3 major groupings, which we can get into later, that we would say are the 3 daughters-in-law of Noah, but the mitochondrial DNA itself, goes back just 6,000 years to a one single female ancestor. But with the Y chromosome, you're saying based on this study, even though the Y chromosomes in the world today, like when we actually focus, let's say, on the variation of it, the Y chromosomes have very little variation, right? But based on that Chinese study and based on the mutation rates in that study, it takes the Y chromosomal ancestor back like 200,000 years, as expected by the evolution model, right?
Can I just apologize for my coughing, by the way? Just some coughing.
No problem.
So, were you continuing your point there, or?
No. I was just clarifying your point that given the empirical method on mitochondrial DNA and given the empirical method on the Y chromosome DNA, you're saying there's a contradiction there.
Yeah, so I'm saying if you're going to say that the mutation rate in the mitochondrial DNA, simple multiplication, constant rate, brings us back to roughly, 6,000 years? Then I don't see what your problem is when we do exactly the same process with Y chromosome, which brings us back several hundred thousand years.
And then, because I typically say that we look at the entire genetic structure as a whole, which speaks to us about the literal Adam and Eve, and you rightfully point to the Y chromosome variation and the mutation rate as expounded upon in that specific paper. Like, I know there was 2 initial papers. One I think it's in 2009 is the one you're referring to with the Chinese men. Is that right?
I think 2009. Yeah, I think it's that year.
And then they did another one in 2015. Actually, before I cover those papers, so in our first discussion, I pointed you to a paper. I can send it to you again after this.
Yeah, sure.
I think I sent it in the comment section, so that could be the miscommunication. But Dr. Nathaniel Jensen, he looked at the Y chromosome, and he looked at all these. I think it's four or five papers that are out on the Y chromosome, and he concluded empirically that we only observe 4,500 years of mutation accumulation in the parental ancestry contained in that record of the human Y chromosome. Did you actually have you read any of the? Is it just the 2009 paper you read, Adam, or was it all of them?
I think it was 2009. Can I ask the point you're talking about now? Is it the one? Because I've read parts of your 100+ falsifications book. Is it that one that's covered in one of the first few bits?
I don't think so, because the Nathaniel Jensen Y chromosome paper was just recent. I'm not sure if we've added it to the book. Matt, can you clarify that?
I did, but it's not the first one. The first one focuses on mtDNA rates, and then the Y chromosome rates are much later.
That's something I wanted to bring up at some point.
Yeah, we can do that. So I would just say, to break it down for the audience too, when it comes to the Y chromosome, all we do in the easiest way is we just take father and son, and then we count how many mistakes occur each generation. This is the empirical method, right? Mistakes happen every generation. The question is, how many? Is it every generation? Is it every few generations? We don't really know until we get say dad's Y chromosome, son's Y chromosome, grandfather's Y chromosome, and do an analyses, and then measure the rate of mistakes. So as Adam pointed out, they did an initial study in 2009 with the Chinese med, and another one in 2015. And Dr. Jensen covers this in his most recent paper. Now, those first two studies, they were based on low quality data and low quality DNA sequencing. So the results that were concluded are what Adam described. He's right on that the rate of mutations that were discovered were slow, which met the expectations of the 200,000 years for the evolutionary Y chromosomal atom. But the important thing is that they did two additional studies, one in 2015 and another in 2017. Those ones, though, those were based on high quality DNA. And what they discovered, which is fascinating, actually, I recommend reading the paper. There's a ton of good sources in it too. They discovered that in the supplemental material, so you're gonna have to look at the details of this paper, but that the number of father to son Y chromosome differences was actually approximately 10-fold higher than previously discovered and expected from the first two papers. So, and they readily admit this as well in the paper, and they even admit that those empirical findings urge them to explore additional filters. I've got the quote here from the paper too. So in other words, they took their high quality DNA results and filtered stuff out that doesn't fit their evolutionary timescale. But the point is those subsequent studies, Adam, that was based on high quality DNA sequencing on the Y chromosome, demonstrates a Y chromosome atom consistent with younger creation.
How are you distinguishing high quality from low quality DNA?
In the study, Dr. Jensen, well, I mean, these studies where they look at the high quality DNA in like the actual secular sources, they explain their methodology. They explain, I guess, the high quality DNA versus the low quality DNA from the initial study in 2009 and 2015. They readily admit that it is, there is a difference in those findings. So I'm not exactly sure.
Yeah, what I mean is, are you saying that if something's considered low quality DNA, does that mean that mutation rate is different?
It may.
What does it mean regarding mutation rates, basically?
So if it's lower quality DNA, then the mutation rate derived from it could be off. That's why it is good to look at if if your first study, let's say on the Y chromosome in 2009, all they had was a low quality DNA, that's really the best that they're going to have at that point in time. But as time goes on, if you get additional studies where now they've got higher quality DNA and all of a sudden they can look at it better than the low quality DNA, I mean, that's kind of just a fact of science,
So
right?
What you're saying is that low quality DNA somehow has different mutation rates to high quality DNA, or less consistent mutation rates from high quality DNA? Because the whole point of this is that we're just measuring mutation rates. So what difference does it make if it's supposedly high quality or low quality?
Well, the low quality DNA that was looked at in 2009 can come up with inconsistent results. So yeah, you're right. They just take, father and son, and then they count how many mistakes occur each generation. Every time there's a mistake, there's a new branch in the family tree. Take those branches back to one male ancestor. How long ago is the question? Well, the low quality DNA sequencing had inconsistent results that were consistent with the 200,000 years for Adam that you're talking about. But in the subsequent studies, the high quality DNA demonstrated that this Y chromosome Adam lived just actually 4,500 years ago. Dr. Jensen proves, and they admit this in the paper, like their exact quote here in the paper, and I believe you'll have to find it in the supplemental material. They say the number of father to son Y chromosome differences was approximately 10-fold higher than the expected number. Considering the range of published Y chromosome copying mistake rates, this finding prompted us to explore additional filters. So they did explore, additional filters in explaining the data, but Dr. Jensen just took that observed mutation rate in the Y chromosome based on the empirical method, and he discovered that this Y chromosome ancestor only lived about 4,500 years ago, which would actually be our nearest Y chromosome ancestor, which would be Y chromosome Noah.
I'm not familiar with any study that says 4,500 years. The minimum date, the minimum time backwards that Y chromosome is supposed to have dated is about 60,000 years, and that's the lowest estimate. We're going to have to move on from this because I don't
want to stick on one topic.
I'm finished. I don't think we're going to get through to the difference between low quality and high quality, because I'm assuming that you think that the low quality and high quality must have both been there from the beginning. So surely it doesn't matter if they're high, low quality mutation rates. But from what you say, should be consistent.
I would just say the high quality DNA gives a better and more consistent mutation rate. And it is also consistent with creationist predictions and actually evolutionist expectations on the decay of the Y chromosome. I know Matt touched on that in our first dialogue. Matt, did you want to jump in here and touch on that? So the Y chromosome is degrading and accumulating mutations at a rate higher than, for example, I think even the mitochondrial DNA. Like it is literally tenfold, as they say, tenfold higher than expected. Matt, did you want to touch on that?
Sure, yeah, but for Adam here, I pulled up the study, and here's how they discovered it. Basically, what it is they said that they filtered variant sites and found that a complete genomic study only kept the allelic SNPs and VQ high. I don't know what that stands for, but it basically has to pass a score to be 95% or better. So the Y chromosome that they had before was of lower quality, meaning what they say in the study is the human Y chromosome is known to have large segments of sequences that are difficult to map until now. So basically, they had a smaller segments of the Y chromosome, not a complete genomic. Now they have 95% or more of the Y chromosome, and it's high quality, so they can get a more accurate study, our trio study. They can see it better. So that's why they got a faster mutation rate. Jensen's the one that mapped it out and found 4,500 years.
Now, actually, real quick, Adam, before you talk, so is it as easy as the high quality DNA that was discovered or used is just a higher range of testing, a higher range of samples to compare versus the low quality DNA, Matt?
Oh, you're asking me. I thought you were asking Adam something.
Well, I'm just trying to break it down for the audience in a more easier way to understand. In a layman way of understanding, is that what it is? There's just a better range of testing and comparisons on the Y chromosome DNA in the higher quality
Right,
version?
exactly. They tested not just more people. They kept testing the Y chromosome database and kept compiling more segments until they had a more complete Y chromosome.
Right, right.
Yeah.
It was just like adding on to the original.
Exactly.
So how much, when Nathaniel Jeanson did this study, how much of the mitochondrial genome was tested?
Asking me that one?
Anybody?
I think this one wasn't the mitochondria at all. I think it was just the Y chromosome.
Oh, the Y. Sorry, Y. That's what I mean. Right, Y chromosome.
Yeah, that's all he was looking at for the study. I posted a video on our channel of it. Unfortunately, it was kind of vague. He said it is in his book, though, so I can get you the specifics on that after. I didn't research it out.
Okay.
If we want, we can just move on. Yeah, I just wanted to show that, like in the mitochondrial DNA and how the empirical method, determines that this mitochondrial ancestor lived just 6,500 years ago or so, and that she would be the one female ancestor of all people. And I don't want to strawman the evolutionists. I know that they believe that Eve and Adam were part of a population, but that's a separate argument, possibly for a separate day to not bog down the debate.
I was going to ask. The general consensus is that even if it was dated to 6,000 years, that doesn't mean that should be the first. Should be part of a population.
Yeah, and that's what I was going to say. I mean, we could discuss that now. The thing is, the out of Africa scenario involving the population bottleneck has some really, really significant problems with it. I mean, it was actually the low genetic diversity that was discovered in the human genome, right? We're all 99.9% similar. That's incredibly low. So and you probably know this, they invented the hypothetical out of Africa population bottleneck to explain the low genetic homogeneity, right? They had to reduce the population. But the thing is, that population bottleneck, Adam, and you can comment on this as well, it would have been at a near extinction level. It would have occurred for a prolonged period of time, and it would have resulted in a severe loss of genetic diversity corresponding to the fixation of bad mutations and severe inbreeding depression. That's the problem. So this inbreeding would have occurred for not one generation, not two generations, but thousands of generations. So that so-called hypothetical population bottleneck of 2 or 10,000 people would have been so genetically compromised that my question to you, Adam, regarding it is, how could such a small, nearly extinct, as I said, genetically compromised population such as that suddenly explode into all parts of the planet, seizing dominion over the world in order to make Adam and Y chromosome Adam coming from a population even feasible?
I'm not familiar with the studies on it, nor any of the science behind it. So if I were to give you any answer, it would be purely off the top of my head. Is the question essentially, how do we get from such a low number to the amount of people we have today? Is that what you're asking? Yeah,
I'm pretty much saying that out of Africa population bottleneck would have been so severely genetically damaged based on the accumulation of mutations from inbreeding, right? Those mutations, those recessive mutations, would have come to the forefront. We can see how implausible that is just by looking at the cheetahs of the world today. We know that they're down to like 6,000 or 7,000 due to prolonged inbreeding. So it's gone on for so many generations that there's been so many bad mutations that have been fixated. So their sperm is degenerate. Conservationists are incredibly worried about the cheetah, and we're not going to see the cheetah suddenly explode into all parts of the world, seizing dominion over the planet. But that's what they say occurred in this out of Africa scenario. So I understand that that's the hypothesis. I'm just saying that there's a lot of problems associated with it.
Yeah. So you're saying there's a problem with, say, if the population was down to 10,000, that would cause severe problems. How then do you explain everybody coming from, firstly, 2 people 6,000 years ago, and then 4,500 roughly years ago, coming from essentially one bloke, Noah, and 3 women.
Yeah, so that's a good question as well. So you are right. You know, there are 2 bottlenecks involved.
Essentially one, because you can remove the first one. The bottleneck is the latest.
Oh yeah, I'm just saying that there's bottlenecks involved in your out of Africa scenario, and then also our scenario. The thing is with our bottleneck, so you got the bottleneck at creation of 2, and then you've got the bottleneck at the flood of 8, I believe it is, and then the Tower of Babel. So 3 essentially, you know, bottlenecks. The thing is, all of them were not prolonged; they were just one generation, and each bottleneck was followed by exponential population growth. And also we explain the vast majority of DNA differences as pre-existing functional DNA differences, as compared to the evolutionist that explains the vast majority, if not all, the DNA differences as a result of mutation over time. So the first bottleneck,
I put in there, it doesn't matter what was originally put into, because essentially you have alleles, and if you've only got 4 people, then you've got 4 different alleles. That's the entirety of variation. So you say that there's this created massive amount of variation, but if you've only got 4 people, then you've only got 4 alleles, potentially.
Right. So actually, that is a good question. I'm kind of glad that you brought that up. So that argument technically comes down to the question, how do we define an allele? So if an allele is defined in terms of a gene unit, then yeah, you're right. Then generating allelic diversity by saying, you know, mutating just one gene per event, mutational event, that would produce almost no diversity. But the thing is, an allele should be defined as a single genomic position. Okay, Adam? And that means that that allele is independent of its relationship to a gene. So that means enormous allelic diversity can be generated not only by mutation, but also by, you know, genetic processes such as recombination and gene conversion, for example. Let me break it down for you. Okay,
yeah, so that's how evolution happens.
Right. But evolution has to explain. Well, here, I just want to break this down real quick, even for the audience, because a lot of that's technical. A single gene, okay, so it typically spans thousands of nucleotides, and single nucleotide variants might be distributed throughout that gene, okay? Let's say, for example, maybe at 90 of the nucleotides within the gene. So if we take the genomic position definition of allele, then let's use the animals at the flood and the ark. Every single one of those single nucleotide variants could have existed in a heterozygous state in each of the individuals of the pairs brought on board the ark. And then at creation, we know that God created in bulk. But I guess going back to the out of Africa and then our bottleneck, yeah. So ours is one generation, no accumulated mutations. So there's no inbreeding problem, because inbreeding involves the exposing or the coming to the forefront of the hidden reservoir of genetic mistakes. But if there is no genetic mistakes, as our model would suggest, then there wouldn't be any inbreeding or genetic bottleneck problem. But for you, like, how would you explain it? If you have all these mutations coming to the forefront for not just one generation, not 2 generations, but thousands of generations, how did this incredibly genetically compromised population of 2 or 10,000 leave Africa, explode into all parts of the world, and then seize dominion over the planet? Like, is there any feasible way of that occurring?
I don't know. I haven't looked at the papers.
Yeah, no, and that's fine.
I'd like to refer you to someone who's actually looked at that stuff, because I haven't looked at it.
I wasn't planning on. I've had like full debates on that exact topic. I'm fascinated with the out of Africa story. We can just. I was just answering. I don't like to straw man the evolutionists. I know that they say that the Y chromosome Adam and the mitochondrial Eve, you know, were part of a population. But I'm just saying that idea is not really supported by the scientific evidence, because you'd have to explain how that would even be remotely feasible. Plus, if these 2 ancestors, as showing here, based on Y chromosome and mitochondrial mutation rates and variation, lived just 6,500 years ago, then we should have plenty of evidence even in written records of some kind of out of Africa type scenario. But I know you had a question as well about the Y chromosome in the book that you wanted to direct at Matt, or because I want to give you some.
I don't want to move on too far from what we're talking, because I do want to say that you're saying that my problem is that I had a genetic bottleneck of around 10,000 people. But you seem to shrug off the idea that you've got a bottleneck of essentially 4 people, because you've got Noah and his sons who are all derived from him, so he's the bottleneck. So Noah and 3 women. So you're saying I've got a problem with 10,000 people so many thousands of years ago, but you haven't got a problem in less time with much less people. So if you're going to give me these problems, you've got to take the problems as well. And when we talk about genetic bottlenecks, it's not just about mutations, it's also about immunity to parasites and bacteria, because if there's only so many people, then what if a virus comes along and infects? If they've all got very similar genes, then they're screwed, essentially. And if we're going to look at this realistically, if you've got 4 people, then you've got a maximum of 4 varieties of each allele. I mean, I know you've talked very vague terms about heterozygosity when it comes. You lost me a bit with that, but do genes come about via mutation? Do we get new varieties of genes?
Well, I'm just saying that to go back to the allele, I'm just saying if you define an allele as a single genomic position independent of its relationship to a gene, which is the way it should be defined, then I'm just saying producing variety according to our created heterozygosity hypothesis wouldn't be a problem. And also when it comes to like the genetic bottlenecks, well, real quick, I just want to address your points. I'm trying to remember what you said. So with the viruses, for one, viruses are beneficial. I mean, you have trillions and trillions and trillions of viruses in and on your body, Adam. You actually have more viruses than you do bacteria and cells, and they're all beneficial. They help regulate the number of species of bacteria in and on your body. In the environment and ecosystems, viruses help regulate bacteria, for example, in the ocean as well. Endogenous retroviruses, we know, are highly functional. So mutation and genetic entropy over time, this all comes back to the genetic entropy model. Adam, because if we started with no mutations at creation with Adam and Eve, and we started with genomes that were front loaded with genetic diversity, there's not going to be any inbreeding problems, not only for that reason, but as I explained, the population bottlenecks were only one generation, okay, one generation at creation, one generation at the flood, and they both were followed by rapid and exponential population growth. So for one, very little of that original created genetic diversity would be lost because the bottlenecks were only one generation each. But the problem with your model is all those DNA differences anyways you explain as mutation, okay? And yeah, like recombination and gene conversion can produce new chromosomal combinations. That's true. I wouldn't say any new genes, but new combinations, new traits can come about through this process. But if our DNA differences are already built in, they're created functional DNA differences, that means new varieties can be created rapidly. Every single generation, especially in creation, in the pre-flood world. I mean, speciation events would have been far faster and more frequent than today. So I think it just goes back to the created heterozygosity hypothesis and genetic entropy that we've talked about that before. We're accumulating 100 new mutations per person per generation. Take that accumulating mutation problem, Adam, back to a point of least accumulated mutations. That would be a point of creation, a point of no genetic degeneration, no genetic mistakes, therefore no inbreeding problems in that first generation creation related bottleneck. Take your time. I know I talked a lot there.
You started to go into genetic entropy there, which I think I talked about in the last one as being a very obscure scientific idea that's only put about in places like Answers in Genesis and this Nathaniel Jeanson, who you quote quite a lot. I've looked at the literature and there's nothing about genetic entropy. And it's one of those things, as well as a lot of other things like mutation rates. It's not just about some specific examples, like you say there's genetic degradation in humans. It has to be across the board. Absolutely everything needs to line up with this, and that's not what we see.
Well, the thing is they found that mitochondria and different things does have different rates. For example, in reptiles and birds, it's extremely fast and it's slower in others. They haven't actually determined a universal clock. They found that some things have faster clocks because they have faster and shorter lifespans, or some will have a faster clock because their metabolism is extremely fast. They haven't exactly figured out the total correlation yet, but standing for truth is going crazy with the science. I just would say that here's a logical question. If you just do the math, evolution says that a bottleneck that happened 200,000 years ago, there were supposedly between 5 and 15,000 women. So if you cut that in average, that's 10,000 different people or 30,000 individuals. Now let's say that if those half were women, again, 10,000, that's the average, but yet we only have one today. Right, one line of mtDNA. That means that 9,999 women either had to die or birth to girls for that to happen. So it seems kind of improportionate, considering evolution said no, there were plenty of other women. There were thousands.
Sorry for my cough. You do not do you not think that they could be part of a population though?
Well, the population,
all the descendants of, say, one person could end up dominating the entire population, or at least all of the members of the population that we've tested.
Okay, here, I'm going to try and break it down real quick, and then Matt, I don't want to jump in on your time, but okay, so it's as easy as this. So over deep time, as evolution would have us believe, is how we got all the species, all the change, of course, all the evolution. Any large population will accumulate massive numbers of mutations, okay? That is how the evolutionists explain the genetic diversity in not only humans, but animal species, for example. Now, this becomes a very serious problem, Adam, for evolutionary theory, because we now know, as I indicated earlier, that the human species has very little genetic diversity, okay? So that observed low genetic diversity is not a problem for us, because we expect that based on the biblical perspective. We came from just two people. God created Adam and Eve. What would we expect? Low genetic diversity, and that's what we see. Now, this was a huge problem for evolution, from the evolution perspective, Adam, because of the low genetic diversity. If we've been accumulating all these numbers of mutations for millions of years, and obviously there's variable degrees of populations, we shouldn't have this low genetic diversity. So they came up with the hypothetical out of Africa population bottleneck. I think they say maybe around 70,000 years ago, which was a near extinction event. So those mutations that have been accumulating over time, okay, they've now come to the forefront. Those enormous numbers of deleterious mutations have gone to fixation. Now, it's not one generation like our population bottlenecks, followed by rapid exponential growth. No, this actually occurred for many generations. That's the only way that they can explain the reduction in homogeneity. But by the time that population suddenly exploded out of Africa into all parts of the world, they would have been so severely genetically damaged that there's no feasible way. I get that it's a hypothesis. I'm just saying there's no feasible way that could have taken place. Just look at the cheetahs today. They're down to 6,000 because of their population bottleneck. Their sperm's degenerate. Conservationists are worried that they're going to go extinct. That's what happens in small bottleneck populations. So that's what we're saying. Yeah, that hypothetical population associated with Eve and Y-chromosome Adam has some really, really, really theoretical issues that I haven't seen anybody deal with.
There's just a few points. Again, you've just bypassed the fact that your bottleneck is in a much worse position than mine, and your excuses are, as far as I'm concerned, baseless and nonsense. And not only if you take one example, humans. I'm not sure on the number that was around in this supposed bottleneck. I haven't looked at any of those studies, so I can't really comment on that. But it's not just a one species thing. You've got to show trends. If you think that everything should have descended from creatures 4,500 years ago, everything needs to align with that. It's no good giving me a specific species that could have gone for a bottleneck roughly around that time. You've got to show a trend. It's got to be a universal thing.
Well, you have to remember that it is a universal thing because, for example, as we say, God created two people, okay? So Adam and Eve. That means that Adam and Eve would have had differences, DNA differences within themselves. And then, of course, this
How do you know
idea.
that? How do you know that?
Well, I'm saying this is our hypothesis, and testable predictions flow from it. That's when we can get in, because if God front-loaded Adam and Eve and all the kinds with these front-loaded functional DNA differences, our prediction would be, okay, the more we sequence the genome, the more we look and study these DNA differences, we predict that the vast majority of these DNA differences, DNA elements, right, for example, the jumping genes, the ERVs, the ALUs, the pseudogenes, these would all play a functional role in the species. But I'm just saying that that's the testable prediction that flows from it. But real quick, I wanted to answer your question of universally. Well, the fact that God encoded Adam and Eve with these front-loaded functional DNA differences applies universally among species, okay? So, for example, each created kind, Adam, was front-loaded with functional DNA differences that, as I've said before, through a variety of processes and mechanisms, such as, let's say, gene conversion, recombination, genetic drift, isolation, migration, whatever, those DNA differences combined with those processes has led to the origin of species. Now, what do we see? Okay, so the Bible says God created animals in bulk. He didn't just create two like he did with Adam and Eve. So we should expect a higher genetic diversity in the animal kinds, in the animals seen around the world. And that's exactly what we see. And then in humans, we see low genetic diversity consistent with that hypothesis. Take your time.
But every animal, well, at least every land-dwelling animal was restricted to either 2 or 7.
Right.
For Noah's. So you're expecting everything, even less genetic diversity in everything that isn't human. So you'd expect surely smaller levels of diversity within animals, but we see animals with incredible levels of diversity. I mean, just look at, we talked about cobras, and I hate to always bring up snakes, but their diversity is enormous just in cobras.
Yes, I agree. So here's the thing, though, okay? All of this to say that the evidence does correlate with, and I understand your objection with the bottleneck of these kinds, and I'll explain that problem too, or an answer to that problem. But this is all to say that the evidence does indicate that God created animals with a greater ability to diversify. In other words, the original animal kinds were front-loaded with more diversity than humans. And this makes sense since we know, as I said, God created populations of animal kinds and then only 2 humans. Adam and Eve. This is why we know the human race has low genetic diversity, which would suggest that we came from a small population. But not only that, like we talked about earlier, which is why it's so important, when we combine the mitochondrial DNA and the Y-chromosome data, it wasn't just a small population, it was a population of just 2, just as the Bible tells us, Adam and Eve. Now, at the bottleneck with the animals, because it was only 1 generation, and this is just basic science too when it comes to bottlenecks, very, very little of that created heterozygosity would have been lost because it was followed by rapid exponential growth. So what we see actually correlates perfectly with this hypothesis. Plus, Matt was explaining in the last debate that study with the DNA barcoding, it proves that over 90% of all life originated at the same time based on the genetic diversity seen in that CO1 gene in the mitochondria. So everything is correlating universally, as you said we should find, and that's exactly what we find.
Can I just nip to the lav? It's my fault for drinking too much cappuccino. I'll be right back.
No problem.
Right, cheers.
Yeah, once you get a scratch in your throat, it's so hard to clear out when you have to talk. It just gets worse.
Actually, that's a good time for an intermission for me too. Matt, I'll let you, I know I've been talking a lot. I don't want to dominate the entire conversation. You take over for a few minutes. For when he gets back, I'm going to run for a quick bathroom break too.
All right, me and Praise. What's going on, Praise? We got to entertain the audience. Uh-oh, you're gone, aren't you?
No, I'm not. 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 Hey, everyone, the chat, the chat, the chat, the chat, the chat, the chat, the chat.
Good conversation, man. Good conversation. Loving it.
You're gonna think somebody's possessed on this channel. Someone just tuned in. Did you see that comment? Look, I honestly have no idea what's going on here.
All right, I'm back. Sorry about that.
That's okay. Standing for Truth went away as well.
Did he?
Yeah.
Do you want to wait for him to come back?
Yeah. Oh, it doesn't matter. Yeah, it's
You
just.
put the pressure on Matt.
Oh, great.
So how are you two been coping with the old COVID?
Oh man, just staying inside. You know, and in the United States, it's real bad. Everybody just goes out anyway, so it's spreading like a fire.
Matt, oh, sorry. I don't know, because I know with what your question, Adam, before we took our bathroom breaks, I'm glad we're all back. Matt, with that study with the genetic diversity, that would indicate the not the creation event, but the flood event, starting with all those animal kinds based on the diversity, correct?
Exactly. That's what that event is actually showing. That's why there was so little barcoding gap between all life. Everything was very short because they expected evolution to be true. They didn't expect to find any gaps really whatsoever. And the low diversity showed that all life was recent and appeared at the same time.
So you mentioned, am I allowed to get back into it now? Are we already set to go?
Sure. Yeah.
So when you were talking there, you said that there's even more genetic diversity in at least the majority of animals, I suppose you implied, from fewer individuals coming off the ark. And you said it was because it was the one generational thing, they maintain the diversity, but that's not how genetics works. If you're restricted to 2 individual animals, then you've got a maximum of 2 varieties of a gene. The whole population doesn't get put into these 2 creatures. It's restricted to 2 genotypes.
Correct. Think about it like this. We say that things were on a family level. So when Noah went onto the ark, he would have brought, say, a wolf and a fox. And so all the species that we've seen since that time has the ability within those original wolf and fox that went on the ark to diversify into the species that we see today. That's what we say. So birds, yeah, a lot more birds went on the ark. A lot more than horses would have. Probably 1 horse pair, because look at how few there are. And they speciate very.
How do you decide?
That's where we kind of get into trying to define what a kind is. Most of the time, it's always been referred to as just something that can bring forth after its kind. That's more along the line of referring to Noah and what went on the ark. And we're trying to make it go even further back. So we're trying to incorporate DNA barcoding with the zygote fertilization and the orphan genes. We try to look more broad spectrum and see what we can actually look at to place into a particular lineage.
So yeah, I'd love to move on to kinds. I think after we've had the bathroom break, it's a nice place to move the topic onto that. So if we're all happy, we could move on to like the definition of kinds and stuff like that.
Sure.
Yeah?
Yeah, I'm good with whatever you want to do.
So I think a good place to start there is for a definition.
Sure.
I know that you gave me 1 or 2 definitions last time, but just to, make sure there's no confusion going ahead from now, let's have it again.
No problem.
Are you asking for a definition from me or Matt?
Oh, either of you. I'm assuming it's going to be at least roughly the same.
Yeah, I figured since this is his time, he can do it. I mean, the definition I always give is organisms that descend from the same ancestral gene pool, because then it's not solely based on reproduction, because we know how genetics work. There's going to be some species that, based on genetic differences, based on divergence, they may not be able to do or be able to reproduce. There may be physical reasons for that, for example, with a Great Dane and a Chihuahua, but we need to look at the blueprint. We need to look at the genetics. I don't want to take Matt's time there. I just heard some silence, so I wanted to jump in. But Matt, take over, brother. Take your time.
Yeah, no problem. No, I mean, that's a good answer. Anything that descended from the same ancestral gene pool would have the ancestry built in from it. We would be able to look at a human, for example, and say, okay, what makes us different? What makes us stand apart as far as ancestry goes? So we say, well, the best way of doing it would be, I really like the DNA barcoding. That's a really good way, but a lot of people don't know about it, so there's no reason to bring it up if you've never understood it. So the best way to explain it would be zygote fertilization, because we know, for example, that a horse and a donkey and a mule are related, but sometimes they can't interbreed, right? But we know that they can in the zygote fertilization. You can do that in a laboratory, and we can see that they're related. Some people have even tried it with chimps and humans. I mean, go back to the 20s and 30s, and it didn't work. So, and these people were...
Yeah, we touched on that last time. Now, the hypothetical I'll give you is that basically the same as last time. You agree that over time, populations will diverge genetically.
Right.
Yeah. So eventually, given enough time, will those genomes have diverged enough that they'll be incompatible?
We think that certain species will be, of course, because like he was saying, like the Chihuahua can't do anything with the Great Dane, but we know through zygote fertilization that they are the same kind. So it's kind of like
that's my point. Given enough time, you've said that genomes diverge over time if populations are separated. If given enough time, if the genomes are so different that they can't even be fertilized in a lab, is that possible?
Well, take a look at the entire canine family, right? We know that no matter where you go with the wolves or the dogs, they can be cross-fertilized. It doesn't matter how many speciation events have occurred. So if evolution was true, we would have an ancient ancestral species canine family that would be so isolated and so different and so far away that it would never be able to reproduce with any type of other canine, but they all can, similar to all cats. Right, we know that if there was a true cat that was like a saber-toothed cat, and it was for some reason it made the ark, and it only lived in Africa, and all the other cats diversified, and there was a house cat now, and it's been house cats don't even go outside, and you're going to say now, well, that obviously has diversified enough that they could never produce offspring, but they can, and they can even graft their kids to one another. So that's how we say if we can identify kind, because that would be impossible if evolution was true, because there would be some ancient ancestor that's supposedly related to the cat that can no longer interbreed.
So you said, well, I just want a straight answer. Is there no way that genomes can diverge so much that the populations can no longer interbreed?
Can they diverge so much that they can't interbreed? I'm not aware of it.
You agree with me that genomes diverge over time, yeah?
Right. Yes.
Will there come a point where those genomes can no longer be compatible?
Yeah, I can jump in. Yeah, of course. See, the thing is, though, if we were to test in vitro, then two species that have diverged from a common ancestor to the point where they're no longer genetically compatible, or there's some type of difference there genetically or physically that doesn't allow them to reproduce, I think the answer is pretty straightforward, that in the wild, in natural settings, they're not going to bring forth, but in vitro type situations, artificial insemination, I mean, there's probably a good chance that they're going to be able to bring forth. Because I mean, that's what evolution says. These populations diverge; they become genetically altered to the point where, boom, there's a new species. Like I understand how evolution works.
So what you're saying is, no matter how much time, if we hypothetically gave 1 billion years to two separate populations, no matter how much their genomes diverge, they'll still be able to be reproduced, to be fertilized in vitro. Is that what you're saying?
I would say that would probably be a reasonable prediction. Like, I mean, you know, I know that they've tried to do in vitro with chimpanzees and humans, as disturbing as that is, and they've had no luck in doing so. But I mean, theoretically, see, here's the thing. So your question is, so our model is the created heterozygosity hypothesis. Okay, so when we look back in time, we're looking at an expansion of the genome. As compared to the evolutionists, they're looking back billions and billions of years to a single-celled organism. They're looking back to contraction of the genome, of course. So that's the big difference. But in our model, okay, in our model, here's the limits, because I think that's what it comes down to. Theoretically, populations may diverge to the point where it's going to be difficult for them to reproduce. That's why our definition is based on organisms that descend from the original ancestral gene pool, not strictly reproduction. But if those ancestors, Adam, and I'll explain this real quick, you can take as much time as you want, if they were front loaded with millions of heterozygous DNA sites, okay, and stick with me, just picture this. This is compared to the evolutionist that says those DNA sites resulted from mutations over time. And I think we're on the same page there, okay? So ours is a different explanation, but this means that the mechanisms by which new species originate can occur simply by genetic recombination and gene conversion. And of course, you have to isolate the population, genetic drift is involved, migration, all these things. I'm just saying mechanisms-wise that can produce variety, because those genetic mechanisms, if the difference is already built in, Adam, they can literally lead to new nuclear chromosome combinations rapidly, even as rapidly as each generation. But here's the answer to your question. Since the created heterozygosity hypothesis suggests that these processes consisted of millions of heterozygous sites, alterations in visible traits and speciation events will decrease in time. For example, if you look at the horse species today, they have an abundance of homozygous DNA sites. We are speciation events are reaching walls. There's less speciation events now because there's less genetic potential, because we look into the past at expansion of the genome that has now decreased. Now we've gone from more heterozygous gene sites to more homozygous gene sites. So if we go 1 billion years into the future, well, that combined with genetic entropy, there won't be any, we'll see extinction. It's the exact opposite expectations of evolution. There is no theoretical billions of years from now in our model. For your model, yeah, maybe, but not our model.
So I think you've moved a little bit away from the main point, which was things remaining within the same gene pool there. How do you explain ring species?
Right. So I'm saying I'm not against ring species. I'm not against rabbits or species of squirrel that have diverged enough genetically, more morphologically. I mean, that's why I brought up the finches. That was a testable prediction that Nathaniel Jensen made that was observed, I think it was 2017. New species was formed based on shifts from heterozygosity, which is what our model says, to homozygosity, subsequent inbreeding, and isolation led to new species. But if with the rabbits, or like the ring species with the rabbits and the squirrels, for example, yeah, maybe in a natural setting they can't reproduce, but we can do tests genetically, whether it's in vitro or if it's DNA barcoding, or we can look at certain taxonomically restricted genes or even function. We can determine if they have descended, which is our definition, from the original ancestral gene pool. Reproduction is a factor, yes, but it's not the only factor, because I do agree some species can diverge and maybe they're not reproducing naturally in the wild. I agree with you on that.
My big problem with your definition for kinds is, I got my Alexa telling me a message. Where was I? Your definition depends on whether things can essentially reproduce or produce fertile offspring, whether it's naturally or within a laboratory. We know that genomes, and I know Rawmat's admitted, that genomes diverge over time if they're separated. We also know that things stop being able to reproduce if their genomes are too different from each other. How then do we define kinds if we know that sometimes these things are no longer able to reproduce, that they're so genetically different that we couldn't reproduce them even in laboratory settings? Because then you cannot define kinds reliably.
Right. So as I said before, my definition, or at least a kind, is groups of living organisms that we can look at. We can determine if they belong in the same created kind if they have descended from the same ancestral gene pool. It seems like you keep going back to just reproduction, but I'm telling you that there's more ways to determine ancestry. I mean, for example, in Darwin's day, his contemporary was Mendel, and Mendel did his pea plant experiments, and he looked at inheritance, Mendelian genetics, for example, recessive and dominant, genes and alleles and things. But the thing is, they didn't even know anything about genetics. So he was just looking at anatomy. He was looking at maybe the fossil record to determine, what's related, what's not. Nowadays, we have genetics, and genetics is where we can determine. It's the only direct method of determining ancestry. A fossil is not passed on, sperm and egg, for example, genes, traits, and genetics. So yeah, reproduction can be a way to determine what a kind is. But if there's been some divergence in genetics, that has, unfortunately, if we can't in vitro everything, and just in the natural, there's some ring species, some species that can't reproduce. Well, now we have to go to the next step, which is, and I don't want to list them all over again, but it would be in the genetic realm. But you need to know that speciation is occurring due to shifts from heterozygosity, so more genetic diversity, to lower genetic diversity, which means eventually you're going to reach walls, you're going to reach barriers, you're going to reach limits. Evolution can't have limits. You're going to go from a single-celled ancestor billions of years ago to all the life we see today,
So what about polyploid speciation then? Where the genome is duplicated, or at least regions can be duplicated.
Right. So yeah, that's another good question. You have all good questions. So I would say gene duplications, for one, are rare events. Secondly, polyploidy. It's like, for the audience sake, to keep it simple, if a lot of this is going over people's heads, have a book. And if you were to duplicate that copy of the book you already own, well, now you don't really have anything meaningful. Now evolution would say, okay, now you're going to get mutations and alterations, frameshifts, for example, in that duplicated gene, or some whole genomes duplicate. But the thing is, that's like adding typographical errors into that second copy of the book you already own. It's still going to be deleterious. For one, duplications are rare. Gene duplications are generally lead to disease, and they're detrimental. So I don't think that that's any help, but it is a mechanism, I agree. But like all beneficial mutations, if you can show me a duplication that is beneficial, it is going to be, it's going to be reductive in nature. So that's my answer to that.
I believe a lot of the plants that we eat are a result of polyploid mutations. For example.
Oh yeah, I would agree. I mean, the thing is, a lot of the plants too, and Matt can speak to this well, especially because this is kind of his forte in his field. But a lot of these heirloom plants, and then a lot of the variations that have been artificially created from them, the heirloom plants are a lot, obviously they got way more genetic potential. That's how come you're getting all these different plants from that heirloom plant, but they're also a lot more healthier in the wild. Matt, did you want to speak to that one?
Sure, if you just think about it like an animal, right? If you take a dog and you take it from a wolf, and the wolf had more variety, and now all of a sudden you create a little chihuahua, if it has a bunch of recessive genes, it can only ever from then on produce more recessive offspring genes. The same thing tends to happen with plants. The only thing is now we're genetically modifying. So now we're twisting things around. But in nature, you're just going to get hybridization. And hybridization is a very similar process, because things will cross, and then their genetic potential gets a little bit dwindled with each time. So that's why heirloom is usually the best, and it's usually the best for people as well. They tend to be a lot healthier as well, because when you start genetically modifying things, people are doing it for flavor. So you get like the cherry tomatoes, which are a lot sweeter. You get corn that can grow in cold places like tomatoes. You're tampering with things, but you're limiting its potential just so it can grow in either a particular condition or taste better.
So what I get from this is that you're saying, yeah, there are these mutations, but they're always negative. Is that what I'm getting from this?
They can be neutral, but most of the time they're limiting in factor, right?
Do you accept that there are positive ones, though?
Right. For example, when we look at the genetic database and we see 6,000 new mutations every 3 months being added, we know that there's a problem, because supposedly there's supposed to be beneficial mutations outweighing these negative ones. And we see every cancer that you can name can be tied to a form of mutation. Where are these mutations that are preventing these things from happening? They're not coming up. They're not anywhere to be seen. we're only getting more damaging mutations being introduced. None of these beneficial ones. All these beneficial ones are being overridden by epigenetic regulation. Every time they think they find a new one, oh, it's lactose. Oh, no, it's not. Oh, guess it's just epigenetic. And that can't change a gene whatsoever, only the way the gene is expressed. So all these beneficial mutations that evolution needs are just wiped out. They're just gone.
Then unfortunately, that's where natural selection comes in. So yeah, there are more deleterious and neutral mutations than there are positive ones. But that's what natural selection acts upon. And if there are even just a few positive ones, they will be maintained within the gene pool.
Oh yeah, no doubt. That's where the theory of evolution comes. You only need one, and one will override the other 20. The only problem with that is experiments aren't showing that. For example, when they went into the Grand Prismatic Springs in Yosemite, they scooped down and pulled up 4 different organisms, and they took these bacteria back, and these are the most primitive ones on Earth, right? And they tested them. They threw them in multiple different scenarios. They threw these in a lower acidic environment, and they tested for beneficial mutations. Nothing happened. The only change was epigenetic. They did the same thing with eukaryotic organisms, because eukaryotes are what we are, right? So they took yeast and they put them in 5 different environments. At the end of 1,200 generations, the only thing that happened is they lost genetic information. All the beneficial mutations were just non-existent, and everything was epigenetic. I like the study so much, I just threw it right into our book and made a video on it. Is incredible, because they need evolution to be true for beneficial mutations, and they can't even find them in the most primitive organisms in these experiments. The only thing that ever goes up is fitness, and that's because of epigenetics.
I can think of a few beneficial mutations off the top of my head. I did mention the muscles in dog eyes that causes puppy dog eyes. That came about shortly after we domesticated the dog. Is that not positive?
Muscles in the eyes. I'm not sure. I have never heard of that mutation.
They are muscles that give the dogs the ability to do the puppy dog eyes, which we respond to as humans. And they're not present in wolves or some of the more primitive domestic dogs like huskies, for example.
Well, muscles can originate, or there's, for example, there's muscles that have been disappearing in the human being. There's one in the wrist. There's one in the neck. There's one in particular in the knee. I forget the name, and humans are starting to lose this one. So I would imagine one day if we start regaining it, or if they found it in people, they'd be like, "Look, we're gaining a new muscle." Perhaps something like that is happening with the dog. I don't know.
Matt, couldn't that be, maybe you can expand on it? Couldn't that even be compared to maybe the Italian wall lizards and their the muscle that was I guess an example of phenotypic plasticity or something to do with epigenetics? Could that be compared to that? Wasn't that involved a muscular enhancement in the wall that allowed them to digest things that they previously weren't digesting before?
Right, exactly. They were vegetarian. They started eating plants, and they needed to slow the process of plant material through their colon down, right? Because they need bacteria to digest plants rather than acid. So this new muscle appeared, and they said, "Wow, look at this! It's never seen this before." But when they went back to the mainland, they actually found that all the lizards that ate plants there also had it. So it comes and goes when it's needed. So maybe these dogs just needed it, and they just started manipulating people with it.
So you're saying the wool? Oh, what was that? Something crashed.
Oh
no problem. It was my kid's toy. Keep going.
So are you saying that the dog kind that was around on the Ark had these puppy dog eye muscles?
I think only wolves and a fox were probably on the Ark, but what do you think, Standing?
Because wolves don't have it.
I would say if those if that information was there for the so-called puppy dog eyes, I mean, I'll have to look into that exact example, but that's why I'm looking at other examples that we do know more of, like the Italian wall lizards. We can see a lot of examples of phenotypic plasticity or just environmental related changes where that was already in the potential of the genome to take place. I mean, are these puppy dog eyes, are they beneficial to them? The muscles?
For manipulating people, yeah.
There you go.
Right. Well, it's kind of like a lot of people say, well, what about blue eyes? That there's a so-called neutral mutation. Blue eyes look good, but the data on blue eyes, for example, suggests, and a lot of people don't know this, that people with blue eyes have more susceptibility to light sensitivity and other eye-related diseases. So usually there is some type of detrimental effect to these changes, and I would predict that if I look into these puppy dog eyes or the muscles that you're talking about that was beneficial, I can almost guarantee that there's probably something negative associated with it. But even if, and I always say this to the evolutionists too, because there are examples that you can give. Oftentimes I'm given like the example of the notothenioid fish that supposedly evolved proteins in the Arctic to keep them from freezing. Examples like this. But the thing is, even if we gave you, Adam, or the evolutionists, one or two beneficial mutations that are truly not deleterious. Picture it this way: if the whole genome is degenerating based on all these, nearly neutral mutations, the detrimental ones, for example, but natural selection, yeah, I can get rid of some of the detrimental ones. But while the genome's degenerating in all these other ways, and while a few nucleotide sites may be improving based on a couple beneficials here and there, huge numbers are still being degraded. So it comes down to a trade-off. I've debated Dr. Stefan Frello, and that's what he said. He said, well, it's a trade-off. It's beneficial in some areas, detrimental in another area. But the question is, Adam, is that trade-off sustainable? Because we can see in experiments, real-time experiments, that let's say Lenski's bacterial experiments, for example. Well, that type of trade-off results in shrinking functional genome sizes. So you're throwing out all this information from lots of nucleotide sites, and then you're trying to replace all that information with one single desirable, let's say a point mutation or a gene duplication. The fact that most beneficials are reductive anyways, the trade-off just won't be sustainable. And we see that in Lenski's experiment. So, I understand that that's the philosophy of the evolutionist. I just don't think that it's demonstrable or empirical.
A lot of what you've said over this last part of the conversation has come down to your ideas of genetic entropy, essentially, which I say again is almost like a plaything of creationist organizations. It's not something that's taken seriously in modern science. So for you to constantly go back to this assumption that genetic entropy is just happening and therefore it explains all these things, well, no, you haven't explained that explanation, so you can't use that.
Well, we've got, a multitude of papers. You can look at the genetic database. I think Matt iterated that earlier, maybe in the other dialogue. You can see how many diseases, thousands of diseases are added every single year. You can see that there's papers after papers of genetic diseases. Cancers are skyrocketing. Even the fact that these beneficial mutations, anyways, most of them are now known to be reductive. There's paper upon paper. Yeah, I can send you papers right now that prove that a lot of these so-called pre-humans, Adam, that the evolutionists will point to as our ancestors or even our cousins, like the Neanderthals, for example, the Homo floresiensis, which is the hobbits, or Naledi, Erectus, Heidelbergensis. I've got paper after paper showing that these so-called primitive humans were not primitive humans. They were highly degenerate modern humans, because it's shown that they're highly inbred due to genetic mutation accumulation. And this inbreeding, this reductive evolution resulted in their reduced brain size, reduced body size in some cases with the hobbits, the pathologies that are seen. Mammoth populations, I got a paper here that shows that they were highly inbred and they carried an elevated genetic load, leading to their extinction. Lenski's experiment, which we can look at today. Viruses, the H1N1 virus, for example, there's a paper, and it's not published in a creationist journal. It's published in a secular journal from Dr. John Sanford and Robert Carter. He showed that the H1N1 virus went from a red hot pandemic to a whimper to an extinction event in 90 years based on a linear accumulation of mutations, genetic entropy at work, he demonstrated. So, I could probably give you 10 papers right now showing the validity of genetic entropy. Take your time. I talked a lot there.
Again, you go into specific species. we need to see a universal trend. You can find any phenomenon in any number of species. So whenever you talk about genetic entropy, you always talk about humans, which for a long time now have essentially bypassed natural selection, which is a thing that gets rid of deleterious mutations and adds positive ones. Well, not adds, but it accumulates them. A lot of these genetic entropy experiments or things that I've seen are on domesticated animals, which go through the same process. So you can take specific examples and say this phenomena applies to that, but that doesn't show a universal trend. You need to show that this is universal for it to be seen as a universal trend. You can't just say we found it in these two, therefore it applies to everything, because that's what you're doing.
Well, mitochondrial clocks are one way that we can do these things, right? So we want to know the mitochondrial rate in different things. Have you ever noticed that when it's pinned up against phylogenetic methods, that they differ by orders of magnitude? A huge difference. Like everything shows that everything should be hundreds of thousands of years old, yet the observed trio studies all get thousands of years. So they're complete contradictory with each other, the observed and unobserved. And
you're saying that mutation rates are inconsistent?
Well, I'm just saying that the mutation rates themselves all show in everything, doesn't matter what it's tested, that it's faster than the assumed phylogenetic method. So that is a mutation rate that they're looking at. That's how they build those charts. When you're looking at what a mutation is, that's one of the ways we can show genetic entropy in name rabbits, alligators, shark, snakes, your favorite. We would look at the mutation rate, and then boom, we have a genetic entropy from that model as well, because it's all faster than the assumed evolutionary model.
I don't think what you said implies that everything has genetic entropy.
Well, if you wanted to test how old a species is, let's say that they want to know how old a new snake is, for example, they might go, well, what's the mutation rate of this new snake? Maybe we can test it. And they look inside of it and they go, well, it's building up 100 near neutral mutations every generation, right? So that way they don't have to look at detrimental or positive. They just test the junk, basically, the neutral ones. And they go, okay, well, it seems to have a compilation of 18,000 total in it. So they knew when that species arose. So that way we can look and go make a mutational rate clock out of that, because if every new generation they acquire 100 new neutral mutations, we go, okay, well, now we know what mutation load is. So now out of those 100, how many are detrimental and how many are positive? Well, they show that as well.
So when I did my genetic analysis of cobras, I put it into something called MEGA, which looks at mutation rates, differences in certain parts of the genome. And it measures all these cobras against each other. There was also an outgroup of vipers. And it measured at millions of years difference. So are there several kinds of cobra?
What was the method you used?
It's just straight DNA. You look at the differences in the genomes, or the genes that we analyzed, and you look at the differences between them. And you take mutation rates of the snakes and you apply that into it. You put it into the test. And it comes out with a phylogenetic tree, which shows you roughly how long ago they were related and in order of relation.
Oh, okay. Well, you said
how they grouped.
I see. So you said it right there. It was a phylogenetic tree. The phylogenetic trees take evolutionary assumptions and add it in. So they assume what happens is the snakes from a lizard, and they believe that this happened around 200 million years ago. So what they'll do is they'll add the mutation rate and extrapolate it like they did.
No, you don't put any supposed dates in there. All you put in is mutation rates and the genetic data.
Well, it seems to be different with humans. For example, when they take...
Well, that's the thing. It's got to be universal. It's got to be everything. That's what I'm saying. You can't just say this specific group have a common ancestor of so long ago. You're saying that everything went through a bottleneck at exactly the same time, 4,500 years ago. Therefore, everything has to have that bottleneck. Everything has to have arisen at that time. So proving that anything doesn't line up with that eliminates it as an option.
Well, that's actually what the CO1 gene barcoding actually did show. It actually did show the little genetic diversity. But you were talking about mutation rates. That's a little bit different than barcoding. It doesn't really look at mutation rates. It's looking at base pair sequence gaps. So it's a little bit different. The mutation rate that you were doing to build your phylogenetic tree, for example, when they were building it with mitochondrial Eve, they said, well, if we just look at trio studies and we know that she's inheriting, let's just say 100 new mutations every generation, and we take that back, it arises just a few thousand years ago. But what we're going to do is, because we know evolution is true, we're going to add the 6.5 million years when we believe they were related to chimps, and we're going to add it in, and that's called the phylogenetic method. Well, the same thing is probably happening with these animals as well. They see the rate and it doesn't match what it's supposed to, so they extrapolate the phylogenetic assumption and into the models, and there you go, you get your millions of years.
So how do you explain my analysis then, where I didn't put any dates in? There were no supposed dates put into it.
I find
we took DNA samples from a whole host of cobras from Africa and Asia, and we compared the genomes. And from those comparisons, the number of differences between them, the estimated divergence time was in the millions of years.
That's strange, because we know that reptiles actually have some of the fastest mitochondrial clocks on Earth, even faster than humans. So if humans are showing to be about 6,000 to 7,000 years old and reptiles are not, that makes me suspicious about what's going on, because reptiles do have the fastest mitochondrial clock inside of them at all. I'll show you the study.
So how would you respond to that then? If
Adam,
the analysis comes to millions of years?
I'll respond to it. So with those snakes, are you doing the phylogenetic analysis based on the gene sequencing of mitochondrial DNA or nuclear DNA?
There were 2 genes from mitochondrial DNA. One of them I know was cytochrome B. That was definitely a mitochondrial bit of DNA. I think there was another one. There was NT3. I could go and find them, but I can't think of the rest on my top of my head. I know that NT3, cytochrome B. Can't remember the other ones off the top of my head.
So you're using conserved mitochondrial genes?
We were using a variety of genes. So the best thing about that is that you can get, instead of just doing one gene where there could be a problem or that particular gene goes at a different rate, if you take a whole host of different genes and analyze them, it gives you better accuracy. So we took, I believe for my experiment, it was 5 different genes, and they pretty much all came up with the same answer.
Right, but it's all mitochondrial genes you're looking at?
No, there were some mitochondrial and some nuclear.
More mitochondrial genes than nuclear?
There were more nuclear ones.
There were more nuclear?
Yeah.
And then how do you explain the origin of nuclear DNA differences?
The origin of nuclear DNA differences would be mutation.
And then how do we explain the origin of nuclear DNA differences?
How do you explain it?
Yes.
I imagine mutation.
Well, no, and that's the thing. Like, we don't want to talk past each other. That's why I always try and clarify with our created heterozygosity hypothesis. The nuclear DNA we're saying was front-loaded. The vast majority of nuclear DNA is the result of divine creation. Okay, so that's why processes like recombination, gene conversion, okay, on these front-loaded nuclear DNA differences can result in new chromosomal combinations quick. So you're assuming in your phylogenetic analyses there that all those nuclear DNA differences that you're looking at are the result of mutation over time. Do you know the mutation rate and speciation rate of the particular snakes you're looking at?
Certainly not off the top of my head, no. I did this study a few years back.
Well, and that's kind of what Nathaniel Jensen's doing, is he's making specific predictions on mutation rates and speciation rates in animals that have not yet been measured. So, I mean, we'd have to look at, like, how many snake species, for example, on this planet right now?
On this planet, a few thousand.
So, I mean, a few thousand snake species from a handful of snake kinds off of the ark is easy to explain. But, like, how come there's not? So, for example, let's say birds and snakes or even lizards. So birds have like 11,000 species, 12,000. Snakes, let's say 3,000. that's consistent with our model, of these species coming from a handful of kinds off of the ark. But according to, let's say, birds, for example, you say birds evolved from dinosaurs, theropod dinosaurs millions and millions of years ago. Why are there not hundreds of thousands of bird species, or why are there not thousands and thousands of snake species?
Because either there's no need for speciation in large populations, or there's a large extinction rate, or a combination of the two.
But then you said in the last discussion and debate, you can't make predictions on speciation rates because there's so many variables and factors involved in it.
Yeah, I know.
So a lot of what you're saying then will just have to, unfortunately, be the result of inference, storytelling. Because if there's no testable predictions that can flow from that explanation, well, then it's not science. Like, I know with mitochondrial DNA, a lot of evolutionists will say, well, natural selection filtered out all the mitochondrial DNA differences that arose in the millions of years to overcome the 6,000 year explanation and empirical evidence for mitochondrial, even mitochondrial DNA. But the thing is, you'll ask them, okay, well, what testable prediction can you make? What's the mutation rate then for the Khoisan peoples, for example? Because Dr. Jensen made a prediction on the Khoisan peoples, and they'll say, well, there's too many variables, too many factors. But where they're saying there's too many variables and factors, we have scientists like Dr. Nathaniel Jensen actually considering those and making direct, very precise predictions. I mean, we'll see if they come true, on the Khoisan peoples in the future, but that means it's science. Science has to deal with, that which is testable and that which is falsifiable. I just think, a few thousand snake species, that's easy to explain according to our model, but millions, and the oldest snake species in the fossil record, how long ago was that? Like, fossilized, like 150 million years or something?
667 million years.
Wouldn't you expect more species then than just a few thousand?
Not necessarily, no.
Because of the variables and factors you were saying. A big point with all this too is, 11,000 bird species, a few thousand snake species, 4,000 or 5,000 lizard species, coming from, or 30 cat species, for example, coming from a handful of ark kinds just 4,500 years ago is easy to explain, especially based on our model of, pre-existing genetic diversity. But yet the evolutionist wants us to believe that all these different species came about from a single-celled organism long ago and far away, billions and billions of years ago. I mean, that is where the imagination lies, it seems. It seems like our model is stronger when it comes to the species.
I disagree entirely.
I did want to ask.
I don't want to go too far from the point, because I don't think I've had a satisfactory answer. How would you respond satisfactorily to studies saying that these animals are they've diverged over millions of years, like the cobras, for example? Are you saying there's more than one cobra kind? You've said
I did explain that.
Yeah, you said the what was it? Hetero. Can you give me the phrase again?
Well, I'm just saying that if you're doing a phylogenetic analysis based on the assumption that those nuclear DNA differences that you're looking at are the result of only mutation over time, well, yeah, you're going to get similar analyses. That's why Dr. Jensen's making specific predictions under the assumption of his model, of our model, the created heterozygosity hypothesis. Now the mitochondrial DNA, yeah, that's based on constant mutation rates over time. So if the snake phylogenetic analyses you're looking at is showing that you said it was the cytochrome B in the mitochondria that you're looking at, and that revealed mutation rates and DNA differences in the mitochondria consistent with millions of years, or was it the nuclear DNA that revealed that?
It was all of it.
All of it in combination.
Essentially, yeah. They all match up. You test them separately, and then you put them. You basically compare them.
Yeah, exactly. So with the mitochondrial DNA, if you're just looking at that and not considering the nuclear DNA in your phylogenetic analyses, what is the origin of the mitochondrial DNA? Is it thousands of years? Is it 100,000 of years?
Millions of years.
If you just looked at the mitochondrial DNA.
Yeah.
I'd have to look at what the actual, because I know that the nuclear DNA, for example, let's say for humans, seems to go back coalescence wise, millions and millions of years. But that's based on the assumption that all those nuclear DNA differences are the result of mutation and not created DNA diversity. The mitochondrial DNA in humans, though, does go back 6,000 years, because we explain the origin of mitochondrial DNA differences for the most part as mutations over time. So if the snakes that you're looking at in this phylogenetic analyses in the mitochondrial DNA alone is going back millions of years, I would just have to see, or even ask you if you know offhand, what is the mutation rate in those snakes you're looking at. I can't make a valid judgment if I don't know we know the mitochondrial mutation rate in humans, which is why we can make direct predictions on human groups that haven't been measured. But what's the mitochondrial mutation rate in the cytochrome B gene that you're looking at in the snakes?
Again, I can't recall off the top of my head. I did it a while ago. I haven't looked at the paper for a while.
Because snakes is your expertise, I'm actually just curious, because I appreciate your knowledge on it. From my understanding, there's two mutually incompatible theories of snake evolution. From my understanding, some say that they evolved from water reptiles, right, mosasaurs,
yeah.
Or others say from burrowing lizards. Like, which one do you hold to?
There's a reason it's very controversial.
That's it. Look, you can take your time and you can have the floor and explain.
I personally suspect they were burrowing animals, much the same way that lizards these days are losing limbs and becoming very snake-like, and they're burrowing animals.
What would the people or the evolutionists that hold to the theory of, because you said that you believe that they evolved from the burrowing lizards?
Again, it's a controversy because nobody really knows. But from what I know, I would go 60-40 in favor of burrowing.
You would, okay. And then the ones that hold to the theory that maybe they evolved from water reptiles, right, the mosasaurs, what would be their disagreements in regards to the burrowing lizards?
I think it's very much lifestyle, and you do also find a lot of fossil snakes that were aquatic. Najash, for example, I believe was. Oh no, not Najash. I can't remember. There's all the Latin names, and all rather complicated. But there were a few fossil snakes from very early on that were aquatic.
Okay, so so you hold to the theory that they evolved from burrowing lizards, then?
I don't hold to it.
Okay, I get what you're saying. So, like, you would say the evidence 60-40 leans more towards that one?
Personal opinion, yes. There'll be a lot of people that disagree with me, but that's what I think.
Okay, so we know, and I don't have to go over all the differences, but we know snakes are very different from lizards. What kind of mutations would you say were responsible for transforming those burrowing lizards into snakes?
Well, the things that really define snakes is not leglessness, because obviously there's a lot of lizards that are legless that you don't call snakes. Most of the differences you'll probably find are in the skull. They've got quite a derived skull compared to a lizard.
Right. And the jaw hinge, like, I know snakes have fangs and a flexible jaw hinge, whereas lizards have, I think, flat teeth and immobile skulls, right?
Yeah, the snake's skull, it's separate on the lower jaw and it spreads out. A lot of people think they dislocate, which is complete rubbish.
What about like the fact, like with movement, for example? I know lizards move by, I think they twist their stiff body or even use their legs, but the tails of snakes are totally different. They're short and they don't break off when grabbed. You said you own a few snakes, you would know that. In comparison to lizards, they've got the longer tails that can break off if another animal grabs it.
Only some lizards.
Pardon?
Only some lizards.
Only so some lizards would they be more comparable in tails to the snakes?
Geckos, for example, are a group that can drop their tail.
But they're still far different than, like, snakes. They use their tails are short. They're mechanically far different than you said geckos?
You get short-tailed lizards as well. Just look at the Uromastyx, for example.
Right. So are you? So you're more so looking to like the skull morphology and things like that versus say
what defines snakes from lizards is mostly in the skull.
Is mostly in the skull.
Yeah.
Because I know like snakes, don't they move? Since we're on movement, don't they move by gliding and they use the bottom scales on their body?
There's a lot of different ways that they move around. Some of them have the S pattern that you see in popular TV, and that they move from their body from side to side in order to have forward motion. Some of them move almost like in a straight line by movements of the ventral scales and the muscles below them. And then you've got things like the sidewinder, which just fling themselves. There's all sorts of different motions for how snakes move.
Don't like one that I'm curious about, like for example, not only do snakes have fangs and a flexible jaw hinge, where the lizards, as we were discussing, have flat teeth and immobile skulls, but you're saying it's in the skulls that we would look at for the defining features. We know some snakes have heat pits on their nose, but then they also have no external ears. Lizards, on the other hand, lack heat pits, but they do have external ears. Like, how would you explain that difference? Is that something that just evolved over time that we can see in the fossil record, or is that something that's inferred to have evolved?
Well, you mentioned that only some snakes have the heat pits, so you've got snakes that don't have heat pits. For example, all the snakes I own, none of them has any heat pits. So I don't see where you're trying to go with heat pits.
Well, I'm just, I guess, going over a lot of the, because even if like some snakes may have this and some don't, or with lizards, with the tails, some may have a shorter one, but there still are these defining features that would need to be explained evolutionary wise, how that burrowing lizard evolved into a snake as we would know today. And it seems like a lot of the evidence, especially from the fossil record, involves things that are lost, right? Legs, for example. So my question would be, how does loss of genetic and anatomical structures to you provide substantial or sufficient evidence for that type of evolution that would say burrowing lizards evolved into snakes over time?
Well, could you repeat that in fewer words, please? I'm trying to get the point of your question.
How does loss of genetic and anatomical structures provide evidence for lizard to snake evolution?
How does loss of genetic information provide evidence for lizard to snake evolution? Is that what you're asking?
Yes.
Well, that's not really the thing we looked at when we said that snakes were derived from lizards. We didn't look at genes and say they've lost genetic information, therefore they evolved from lizards. It's different things.
I know, but what I'm saying is, in the fossil record, for example, what we look at is snakes. You're saying that they evolved from lizards, but then the fossil snake species we see that actually have like those, I guess, hind legs, you could say. It looks like the further back, the furthest back you can go, the snakes you find, they don't have any legs. And then you can find some snakes with legs 50 to 100 million years later.
Yeah.
And then, but it should be like the reverse. Like, why, I guess, based on your reasoning, and because I imagine that the differences we're talking about and how those changes would be required, we can infer from the fossil record. But snakes, so you're saying snakes would have gained legs, and then like I see reversals in the fossil record. Like, how do you explain that?
This is a fundamental misunderstanding of how evolution works. You get a population of snakes. Now, the original snakes would have had legs because, as I mentioned, leglessness isn't the defining characteristic of snakes. It's in the skull. So the original snakes would probably have had legs. You would then have the divergence of these big groups of snakes. You find it when there are adaptations, there's usually radiation where you get masses of groups of these animals and they spread across large areas of land, maybe even across the globe. So back millions of years ago, say 200 millions of years ago, you probably would have had snakes in various parts of the world with legs. Over the next millions of years, some of those groups of snakes would have lost their legs. Some of those snakes might have been fossilized, and that's like the ones you see in the fossil record. So you've got legless snakes showing up in the fossil record. You've still got snakes with legs living elsewhere in the world that either went extinct later on or lost their legs later on, And you still get some of those legged snakes moving on through the years, and to the point where, say, 20 million years ago, you still had snakes with legs. It's not the snakes didn't all evolve as one single population. The snakes would have radiated as as snakes with legs into multiple populations. Some of them would have become legless long before the others, or some of them may never have stopped being legless and just gone extinct and left only the legless snakes. So to have the earliest fossil snakes we find to be legless and some legged snakes to be found later on in the fossil record isn't a problem at all.
Right. So it's just because my point is, like, according to our model, of course, and as I've said many times, it's genes that are inherited. I think genes is where we can directly determine, what's related, what's not, the origin of these traits in regards to, let's say, the lizards to snake evolution. We do see snakes with no legs. We see snakes with the the hind legs, for example. But according to our model, those layers are not, they are not absolute ages of time. So it looks like the evidence would suggest, either the snakes with the hind limbs are used, as we can see today, for copulation, reproductive purposes. I think for clasping during reproduction. Like, would you agree with that function, or would you say that they're completely worthless, the hind legs?
Trying to think, because there's quite a lot of groups of snakes that still have it, and I believe it varies between species.
Because I'm trying to, like, the ones you see with hind legs in the fossil record, they could either be, like today, used for reproductive purposes as a function, or like, for example, Najash, is that the name of the
two-legged snake? Yeah, it's got two hind limbs.
Isn't that and that's a genus of snake, right? Najash, like there's many species in that genus.
Yeah.
So that snake has the hind limbs on the side of its body, though, right? Versus it's coming from the side of the body, isn't that? I remember you pointed that out before.
Yeah, that's you won't find lizards with legs below. They're always
right.
Side.
Right, and that's like a defining trait in that in the fossils of the genus Najash that you would point to that say, okay, here's your two-legged snake. That would be your transition between your burrowing lizard to your snakes with legs.
Not necessarily, no. Because as I said, leglessness is not the definitive characteristic of snakes. It's in the skull.
Right. Are you familiar with I think it's the HA1 gene for back legs? It's in a non-functional or degraded form in snakes. That's why sometimes you can see snakes that are born with these useless legs that don't actually function because that gene is broken, suggesting that there were some species of snake in the beginning. Like according to the Bible, right, the serpent was cursed. And that serpent would have had legs. And if God, through epigenetic purposes or just, the damaging of genes, like we see with this HA1 gene, it was a process over time that these snakes would lose legs based on the degradation of that gene, for example. I mean, that's consistent with our model of snake species with some legs like Najash, for example, that have lost them over time, and snake species like the furthest snake species we can see in the fossil record that have no legs at all, or just basic hind limbs that sometimes evolutionists will say are vestigial, but they're actually used for reproductive purposes. So that would be, I think, pretty consistent with our model. Take your time. Yeah, go ahead.
It's interesting what you brought up there. So God would say, be gone with your legs, snake. How did it work there? Was it instant or was it over time?
Good question. Yeah, I'm proposing theoretically that it was over time, because you can see that specific gene for legs, for the back legs, is degraded and it is in a non-functional form. So if God, because it says God cursed the serpent, so if those genes were, because you've got Garden of Eden, you've got the serpent, but you've got also outside the Garden of Eden, you've got snake species that theoretically, like Najash, would have the hind legs, that they would lose them over time. Yeah, I don't think they lost them right away, no.
Do you think all the fossils of snakes were caused by the Noah's flood?
I would say that the majority, like for example, the ones that are found 100 million years ago or 150 million years ago, for example, like with Najash. What's Najash? 100 million years ago that they've discovered that one? Because you would say that snakes evolved from lizards 200 millions of years ago, right? Was it during the Jurassic period, maybe?
It was around the Jurassic, yeah, I believe.
Yeah, okay, so let's say
the oldest fossil snakes we have are that long, but the genetics, yeah, goes back further, obviously.
yeah, okay, yeah, then yeah, they would have been fossilized during the flood. That's right.
So there are some snakes in the fossil record that have legs. Some have 2 legs, some have 4 legs, some have no legs. So after God punished them, some of them lost their legs at quicker rates than others.
Yeah, that's right, because I'm saying that God would have cursed, if they're going to lose their legs, and we see that there's this gene, what is it, the HA1 gene, I think it's called. Hope I'm not butchering that. I can look it up. But that is in a functionless form in snakes. We can see that today, sequencing their genetics. The snakes we see in the fossil record, they're just bones. So, you know, we can infer the best we can, but we don't have any genes. We don't have a bone, like I said, it's not passed on sperm and egg. I think that's an appropriate inference, that there would have been snakes with legs at creation. We see that the serpent had legs, and you know, he was cursed. If it was a genetic curse where over time, and like you said, yeah, maybe some snake species lost it faster than some based on environmental conditions, because we know that a lot of change is environmentally dependent. So I think that that's perfectly consistent. Yeah.
Were certain lizards cursed as well then?
Good question. Yeah. So over time, we can see that there's certain lizard species right now. Are they the skinks, for example? I believe that they are burrowing lizards and they are losing their legs.
Yeah.
Yeah, the thing is, the curse at the very beginning of creation in a world with no accumulated mutations versus today in a world with accumulated mutations, legs, as you've said, or fish, blind cave fish, they would be prone to losing traits or features such as that. So yeah, I would just say that loss of anatomy, loss of genetic information is consistent with our model of genetic entropy and reductive evolution.
I will say it's hardly a curse, though, because snakes are one of the more successful species on the planet. Their leglessness has allowed them to take advantage of a whole host of environments. They can be arboreal, they can be terrestrial, they could be subterrestrial, they could be aquatic. Their leglessness has allowed them to radiate to loads of different areas, and they're very widespread. Would you still consider it a curse, even though that's the case?
So would you like, in your next life, would you like to come back as a snake that has to crawl around on its belly eating dust all day?
They don't eat dust.
Okay, well, answer the question though. Would you? Okay, well, is there a disadvantage at birth when you get that snake that is just born and has to crawl around on its belly? I mean, in the world of predators and things like that, isn't there a disadvantage there? I mean, I agree with you that any kind of species is going to be able to learn to adapt to its environment, of course, and you can even see that in like Lenski's bacteria, for example. But when those bacteria are adapting to their artificial environment as well, and yet we can see that they're doing it through lots of function, lots of traits, lots of information. So I mean, yeah, I'm not against adaptation if that's what you're getting at.
To answer your question, if I was a snake, I wouldn't mind, because that's how snakes live.
Right. Okay, go ahead. Because you're a herpetologist, I wanted to give you some considerable time discussing snake evolution. I mean, that's your field. But I did want to go back to one point before I forget it. I keep trying to keep it on the back of my head, or in the back of my mind, from when you were discussing with Matt, because you were talking about how like a universal genetic entropy. If we can see it in viruses, okay, the H1N1 virus, which has high generation and high mutation rates. What about in the long-term E. coli experiment by Lenski, which I mean I've mentioned many times, so I'm sure you're familiar with. But that work does show that, for one, the rate of beneficial mutations is less than 1 per million, and then that experiment clearly showed that most of the documented beneficials were loss of function mutations. And I think that that's a perfect reflection of what we are describing as reductive evolution or genetic entropy. So if we can see it in humans, which you say there's a problem with because lack of natural selection, okay, fine. We can see it in mammoth populations. We can see it in bacteria. We can see it in viruses. We can see it in inbred populations, of course, because then that's just going to speed up the degeneration process. We can see it with the accumulation of disease. Like, is that not universal enough for you?
No. Because we've got examples where the genome isn't decreasing.
In what?
Well, for example, polyploid.
Right, but
that's increasing genetic information. The dog's one, the puppy dog eyes again. That's caused by a change in the genes, which results in these muscles, these new muscles.
Are you saying that wolves don't have these puppy dog eyes or the muscles associated with them in a much more degenerate form?
They don't have the muscles.
I think they do in a degenerate form that have been amplified in artificially bred dogs that have now served a benefit to some kind, I believe. No?
No.
With gene, you'd have to look into that, but that's I believe that the wolves do have it in more of a degenerate form, but we can look into that after. But the gene duplications that you're referring to, like I said earlier, are universally deleterious. There's no, you can have 10 copies of the same book and start introducing typographical errors in those extra copies of the original book. You're not going to really get anything new and meaningful. I mean, typographical errors indicate mistakes, and that's what we see with mutations. Plus, duplications are rare. I mean, I pointed out earlier, like, is there, other than natural selection, and based on the known functionality of the genome, the known functionality of all the genomic elements, including the pseudogenes, ERVs, the various classes of retrotransposons. I mean, they're crucial for genetic health and function. If the genome, the genetic information is nested and it's overlapping, any small little change is going to be not neutral. It's going to have some type of deleterious effect, even if it's invisible to selection. That's the point. What type of selection can you give me that's going to be able to filter out those nearly neutral mutations that are only just very subtle on the genetic content?
Natural selection.
So it's only so. But if natural selection, Adam, is only acting upon the most detrimental of mutations and amplifying a few really beneficial mutations, well, what's it going to do against the worst kind of mutations, which are the nearly neutral, very subtle mutations that are just very slightly deleterious? What does it do against? It can't see those.
If it's very slightly deleterious, then it is deleterious.
Right, but it's like a book the size of an encyclopedia. You get one single spelling mistake on its own, it's inconsequential, but the buildup of them over time degrades the message, degrades the book, and that's what we see with these nearly neutral mutations. If the genome was truly 90% junk, you've got 10 mutations that are deleterious entering the human population per person per generation. But we now know that that's false. We know that the genome is a lot more functional than that. So therefore, you've got many more deleterious mutations accumulating per person per generation. Natural selection is only going to get rid of the worst. The ones that are very subtle to fitness, like that single spelling mistake in a book the size of an encyclopedia, they will build up. That's why we see the genetic disease. I mean, you got to have a type of mechanism, or else it's just you're just hanging out in the realm of philosophy.
Okay.
Well, let's do this, because actually we went for a couple hours. It's been a lot of fun. My kids have been up for a little while. You're the guest here. Actually, there's a few other things I wanted to discuss, but for sake of time, maybe we can do a third one, because there's so many points to touch on, especially with kinds and speciation. Let's give you a few moments, even a minute or so, to kind of sum up what you think, maybe address some of the points you feel like are hanging. Adam, take your time.
I've enjoyed myself tonight, so thank you, all three of you, for being here and having this discussion. It's good to do this sort of thing when, let's be honest, we can't really do anything else. The main thing I'm taking away from tonight is that. Oh, thanks.
Attaboy, praise.
We haven't heard enough of that whatever it is that Praise is playing with. A lot of it has come down to genetic entropy. And as I've said a couple of times, that's simply not an idea that's taken seriously in modern science. And because everything seems to fall down to that, and this idea of created heterozygosity, which is just asserted without anything backing it up, the fact that I'm told I have a problem with a bottleneck of 10,000 when the opposition has a bottleneck of 4, it's a lot of the stuff is just it strikes me as very vague, very much an exercise in cherry picking, and quite frankly, you haven't convinced me.
Is it okay if I ask you one question before we end it? Like with our model, right, the created heterozygosity hypothesis, if we're saying that the new species are going to arise through shifts from more heterozygous nuclear DNA to more homozygous nuclear DNA, which is what we've seen in these new finch species, for example, that would mean, according to our model, that those new species are effectively losing a number of those heterozygous DNA variants that were front-loaded in the created kind. So my question in regards to that would be that if according to our model that speciation potential lies within the ability to spawn genetically and visually diverse offspring based on heterozygosity levels, would high heterozygosity, wouldn't that indicate higher potential for speciation, while lower levels of heterozygosity would represent a lower potential to speciate? Would you agree with that?
Speciation is very much dependent on what's happening with the population regarding its environment. It's not necessarily down to the makeup of the genome. It's whether group or populations are isolated from one another and their genomes allowed to diverge.
Right. No, and I agree with that, but I'm trying to say, because you're like a lot of it, I just want to make sure that you understand before this conversation ends. You're asking like, well, where's the limits, or what happens in billions of years from now? But high heterozygosity levels would represent a greater potential for change, for allelic variety. Would you agree with that? Higher heterozygosity would mean higher potential for change in speciation, right?
So you changed things there. You changed it from allelic variation to speciation.
Well, because the higher allelic variation you have, the greater potential for change, the greater potential for speciation. If the process of speciation involves shifts from heterozygosity to homozygosity, the lower heterozygosity you have, like low heterozygosity as compared to high heterozygosity, would represent a lower potential for speciation and change. And why do we see in species today, like the horse species, for example, they have more gene sites that are homozygous versus heterozygous? Take that back to like the horse kinds off the ark, then they would have more heterozygous gene sites, and take those horses back to creation. There's your pre-existing heterozygosity. Don't you see how it's a downhill change? Like, we're looking back in time to the expansion of the genome, but evolutionists, it's the opposite. You're looking back to a single-celled organism billions of years ago. You're looking back to a contraction of the genome, because from a single-celled organism to now, fish, amphibian, reptile, mammal, monkey, bird, or monkey, man, that would require the expansion of genetic information versus us, the opposite.
How do you know that?
Well, if you started off with a single-celled ancestor,
how large was the genome of the universal common ancestor?
Well, you tell me. I don't know. It's your story.
Exactly. We don't know. So for you to say that I've got a problem because we need to add more information from the original common ancestor, you've jumped to that without actually providing a reason to.
Well, no, I'm not necessarily saying you have a problem. I'm just saying, for clarification purposes, do you not see that it's opposite expectations? Because we're looking back in time. We're looking at an expansion of the genome. We're looking back to the creation of these kinds and Adam and Eve that were front-loaded with all these pre-existing functional DNA differences. Over time, through genetic processes, recombination, gene conversion, isolation, divergence, genetic drift, all these things that lead to speciation, we're now looking at the contraction of that originally expanded genome. right? Because you're asking about the limits. Well, I'm giving you an answer to the limits. I just want to, especially for the next conversation. That's why we reach walls or barriers in speciation potential, because those gene sites that were heterozygous, we're losing them over time.
I think you'd need to provide papers on that, though. I think you're getting that from the idea of genetic entropy. Is that true?
No, I mean genetic entropy, because remember, we explained the vast majority of DNA differences the result of created DNA diversity. But the genetic entropy model would say, yeah, we're accumulating mutations since creation. But evolution says all those DNA differences were mutation. I'll tell you what, I do have papers on that. I'll send those to you. You can look them over. And then the next time we have a discussion, we could, I guess, expand on this conversation where we left off here.
Sounds like a good plan.
Okay. Did you have any final words before we?
I gave my final words.
Okay. Well, I appreciate you giving me the chance there to ask another question. And yeah, like you, I really enjoyed this conversation. I enjoyed discussing snake evolution. We touched a number of really good topics. So I hope the chat had fun. Matt,
Do you understand my point? I just want to clarify, because I know this is in your book. Do you understand now that because we find legless snakes in the fossil record before legged snakes, that's not a problem?
well, I know according to like evolutionary theory, yeah, you guys would look to like loss of traits evolution, convergent evolution, things like this to explain why you would find snakes the furthest you go on the fossil record that don't have legs, and then you'll find snakes with legs. Yeah, it's just like Tiktaalik, for example. I'll point out that you found tetrapod footprints in Poland that predate Tiktaalik.
Yeah.
Now, I understand that Tiktaalik, you'll say, it doesn't necessarily mean that that was the first transition.
Yeah. That's why we have things as sister taxa rather than putting things as nodes.
Exactly. Yes. No, I agree. I agree with like the evolution, I guess I want to say story of it. You know, I understand that. Yeah.
That's my final word. Then I'd like to see that correction in your book.
I'm not saying that. Like I'm just saying I understand the it would be almost like an evolutionist saying, like, you do understand why, you know, we expect dogs to produce dogs based on the law of monophily, right? We wouldn't expect a dog to give birth to a cat. That would be me like saying, well, no, I understand that it's genes and traits that are inherited, and of course, based on the law of monophily, you know, eukaryotes are always going to produce eukaryotes, mammals are always going to produce mammals. I understand that. I don't agree with it supporting evolution. So that's kind of what I mean. Like, I understand what loss of traits evolution is. I understand what convergent evolution is. I just don't believe that it rescues you from the problems.
Okay.
I hope that makes sense. But I do understand like what you're saying. Yeah, I agree that makes sense in the evolutionary worldview. But remember, we don't look at all those snake fossils in the geologic column, and we don't look at that column like it's different ages, millions and millions of years. We believe that it was all created in one year-long worldwide flood. So, but we can talk about that later. Yeah.
Right. Okay.
Okay. Yeah. Awesome. I appreciate it. I hope the audience had fun. Matt, hopefully you didn't fall asleep while Adam and I were blabbering on there. You can say final words, brother, and we'll shut her down.
No final words. It was good. I'm just here to chime in.
Awesome. Awesome. Adam, like I said, it's always a good time. I hope you always feel like you got enough time to talk and that it's respectful. And we'll touch on a few things in our next discussion. So we'll hand it over to Praise for his final words, and he can shut it down.
Yeah, I had a blast. It was a good discussion. Thanks, Adam, for coming in here, and SFT and Ron, Matt, and everyone in the chat. Thanks for your super chats. Thank you for engaging with us. Hopefully we do this again. I don't know if we're going to be on tonight again or not. You guys know, or?
I have no idea.
I think we'll coordinate afterwards and see.
Yeah, we'll see. Anyway, we're going to say God bless and see you later.
God bless.